textbook also, every textbook says that. The father of genetics. The father of genetics. I hate that term. It's a load of hooey. It doesn't matter. The reason Gregor doesn't matter is because Gregor published in a journal that nobody read, and nobody understood what he said because he was like way ahead of his time. Gregor Mendel, in truth, does not matter at all. Because Gregor, everything he did was forgotten by science for 30 years until someone else rediscovered all of it. A guy named Conrad de Vries went and rediscovered everything this guy discovered in the first place. Everybody talks about Gregor Mendel. Nobody talks about de Vries. But de Vries, he rediscovered it. He found all the same stuff and more. He's the one who discovered mutations. And that's the only reason we even know about it. So yes, Mendel was first, but because nobody would even know about it if de Vries hadn't made a big to-do about it. Mendel studied peas. He looked at 7 different traits in peas. He looked at mathematical patterns. Before Mendel, everybody said, oh, kids are a blend of their parents' traits. For most traits, that's not true. For most traits, you have one that is dominant. For many, many traits, you have a dominant and you have a recessive. And if they both give their genes to the baby has both genes. Do we agree? But do you see both genes? Because the dominant is blocking the recessive. This has the gene to make white flowers. You're not seeing white there, right? If you have complete dominance, the dominant will fully block the white and you won't see it. You might as well write this down. These are definitions that I would recommend knowing. We're going to use them through the rest of the next 2 PowerPoints. The dominant is a version of the gene that will be expressed if it is present. Expressed means it will be seen, it will affect the organism. The dominant will be expressed if it's present. The recessive will be expressed if it is not being blocked. Okay? So here we go. He read pea plants that had purple flowers with peas that had white flowers, and in the first generation of kids, we call that the F1 generation, he knew they had to have the information for white, but he didn't see it. But more importantly, they are all purple. It was not what everybody had said would happen. Everybody said, oh, kids are blending into parents. There's no blending there. He didn't get some washed out halfway purple. And they're not some washed out purple that's halfway between the two. This purple is as purple as that purple. Does that make sense? He didn't get some washed out halfway in between the light colored purple. His purple was as purple as that purple. He had a pure breeding purple plant from a line of pure breeding. It took him a year to set up. I don't know, maybe more than 1 year. But he had a line of pure breeding purple plants that only he knew that their parents were purple, and their parents were purple, and their parents were purple, and their parents were purple. He did the same thing with the white flower ones. Then he took one of those purebred purple ones, these purebred white ones, he breeds them together, and you don't get some washed out halfway between. You get purple, just as purple as that. Because the dominant trait, the purple trait, is what you express. And obviously, you can breed a black cat and a white cat and a white cat together, you get all gray kittens. But it's never been that way. All right. So one of the things that Mendel figured out, one of the big ones, was that kids are getting 2 sets of information. They get a set from dad and a set from mom. You get a set from each parent. And for most traits, they don't blend. He said they don't blend. But we'll get to that. Anyway, he didn't know what the information was. He didn't know how it was stored. Remember, they discovered the structure of DNA and how all that stuff worked in the 1950s. And then de Vries rediscovered it. But you get my point. This was a big deal. It was like the information to make a white flower is being passed on even if you can't see it. That was something that nobody understood could happen. And And he figured it out. And then, of course, the other two dudes figured it out too. Words and drive. Anyway, so we have words for these. He calls the one that you see the dominant one. And then the one that you're not going to see, the recessive one. I'm going to have you write down some definitions right now. You'll see them again later. If you're going to be a lazy butt, write them down when we get to that slide. But write them down now, because I'll use them the entire chapter. And they are not the same definitions as the book. They're better, and I mean that. The definition for dominant is the gene that will be expressed. That means the one that actually does something that you actually see. The gene that is expressed if it is present. That's a dominant. The definition for the recessive is the gene that will be expressed if it is not being blocked. Are you with me? Andre, repeat them.
Dominant is the gene that will be expressed if it is present.
Yes, yes.
And then recessive is the gene that will be expressed
if it is not being blocked. So let's take a look at this. We've got two genes, and I want you to edit that slightly. For dominant, I want you to say, instead of the gene, say the version of the gene. This is important. I'll explain why it's important in just a second. And for recessive, say the version of the gene. Okay? Because this is the same gene. Okay? There's one gene right here for color. But there's a version that makes you make purple pigment, and there's a version that makes you make the white pigment. Does that make sense? Alright? So the dominant is the one that will be expressed if it is present. Would you agree he passes on information to make purple flowers to this generation? Would you agree this one passes on the information to make white flowers to this generation? Right? So this has two different types of genes inside of it. Do you agree? Alright. There's a science word. We'll get to this word later. You don't have to write it down now. You'll see in a second. But for any time you have two different versions of a gene, we say that you are heterozygote. Hetero means not the same, right? Right? Homo means the same. Hetero means not the same. So what we have here is a heterozygote flower. It has the dominant allele, dominant version of the gene. It's got the recessive version of the gene. Good. And what color is it? Purple. Because purple, what's your definition? Purple's dominant. What's your definition of dominant? Will be expressed if present. Would you agree? It's present and it's expressed, so you look purple. Now, what was our definition for recessive? First of all, is the gene present? Yes, the white gene is present, but it's not expressed. And the reason is our definition of recessive was the recessive gene is the gene that will be expressed if it isn't being blocked. The dominant is fully blocking the recessive, so you don't see it, even though you carry it. This is a carrier for the recessive white, and you'd never know looking at it. Now, if you went to a sample of a plant and did a genetic test, you could find that allele, that version of the gene. But if you're just looking at it, you'd never know. Am I right? All right? So what happens? You breed two of these together, and you're going to end up with a 3-to-1. And then, when he breeds two of these together, the next generation, the white comes back. No, that was a big deal. Nobody understood that until he did it. Of course, nobody understood it afterwards because nobody paid attention when he did for 30 years. What he was looking for was mathematical patterns in offspring. Remember I said memorize the magic ratios? Memorize. I want to memorize the magic ratios. All right. So Mendel's looking for patterns, ratios, things like that. Before Mendel, everybody said, oh, well, kids are like a blend of their parents' traits. And Mendel's big breakthrough is that that was false. Now, after Mendel, then we came up with fancy words like genetics. And Watson and Crick discovered the structure of DNA. We already said they stole part of the info to help them. But we figured out that information was stored in DNA. We didn't know that until the 50s. We have a whole new field of science. The big current trendy thing for the last quarter century. When I say new, I'm talking about something that's only been around 25 years, which means it's just starting to show up in textbooks, is epigenetics. The idea that it's not just what DNA you have, it's what you do with it. Which genes you turn on and which genes you turn off that matter. And it's still too new to show up in most textbooks. If your textbook doesn't have it. That's called epigenetics. And we'll talk about it a little bit, but it is the idea that what genes you have are not as important as what genes you turn on or turn off. You can take a gene that you have and you can wind it up really tight and put methyl groups on it, you wind up around the piston, and then you're never going to read it again. It's like you didn't have it, you turned it on. That's a big, big deal. Anyway, so yeah, we now think that that controls just as much as what genes you have. Let's talk about why. Break out your Punnett squares. Let's talk about why. All right. I'm so prepared here. We're gonna go.
Is that always true? Is it just for flowers?
Very good question, and the answer no, it's not just for flowers, and the answer no, it's not always true. There are other forms that we'll talk about in just a little bit. But work with me here. First, I'm going to draw what we had. Would you agree that each one of those flowers, would you agree that each one of these flowers has a purple gene and a white gene, right? I'm going to use the letter C for color, because we're talking about color. Do you agree? You can use any letter you want. But I'm going to use C for color. So, would you agree that the dad is going to have a big C and a little c, right? A dominant and a recessive. Can you see that? You see it? All right. Would you agree that the mom is going to have a dominant and a recessive? Because they were the same, right? Right? We were breeding two of these, so they're going to be the same, right? Well, here is one of the most common things you see in a genetics problem. If dad can give a big C or a little c, and mom can give a big C or a little c, what's going to happen is that if they both give a big C, you have big C, right? There's a word for that. That's the dominant gene, right? We would say that's homozygous dominant. So what do you think we'd say if they both give their little c? Yes, exactly right, Lisa. We would say it's homozygous recessive. And then these two will be just like their parents. They get one big and one little, and we'd say they're heterozygous. Right? So, tell me, what color is that going to be? White. Because our definition of the recessive is the thing that will be expressed if it's not being blocked. There's no dominant there to block it. It's all recessiveness. So it's going to be expressed. Do we agree? All right. This, there's no recessive gene. It's just the dominant gene. So what color is it going to be? Purple. And these are going to be, well, but for complete dominance. We're talking about complete dominance. We're talking about, I know you're getting ahead. We're talking about complete dominance. We're talking about those purple flowers. So they're going to look purple too. Do we agree? Because the dominance is blocking the recessive. You end up with 1, 2, 3 that are purple and 1 that's white. My God! 3 purple, 1 white. He figured that out. What he was looking for was mathematical patterns and all. That's what he figured out. Now, he didn't invent Punnett squares. Punnett did. He probably figured that out. But he figured out the ratios, the magic ratios that you're going to memorize. And that is, we'll go back to it, but that is, thank you. I wish I'd show the Punnett square. There's the Punnett square. That's the Punnett square that I just drew, right? This is the one that's in every flipping textbook. You have a big and a little, they use P, but you have a big and a little and a big and a little. If they both get the big, you're done with that one. If one gets big and little, then the big, the dominant's going to be expressed. The little will be carried but not expressed. But if you have two littles, two recessive, if you're homozygous recessive, bam, you're going to look like the recessive. Because you can't look like the dominant because you don't have the dominant genes, right? Right? Easy enough. All right, so let's get back to we'll get through these in order. We have to go through a lot of nerd words to do this. All right, so is everyone good with this? Yeah. So Mendel figured this out. He figured out that the kids were not a blending of the traits, that you could have a trait that fully blocks the other trait. That was new. That was news. Nobody knew that. And unfortunately, 30 years before he found it out. but, yeah, he found it out. Okay. So Mendel figured out that there were two sets of information that came from the parents. One from dad and one from mom, right? He figured out that most of those traits don't blend in any way, that instead you'll have one that blocks the other. Good? Now, Mendel didn't know that that was DNA that was doing that. He didn't know it was stored in the DNA. We didn't know that until the 50s. Yeah, he had no idea. But he knew that some information was being passed on, and it came from both parents. And that you didn't lose information just because you couldn't see it. That was a breakthrough that nobody paid attention to for a while. All right. After Mendel, that's when we came up with the word genetics. What we're talking about is genetics. But Mendel didn't even know that word. He didn't use that word. And so, diploid cells, this is just a recap. You know this already. Diploid cells are most of the cells in your body. They have two copies of the genes. You get a copy from dad, you get a copy from mom. And so 23 from dad, 23 from mom, 46 chromosomes total. Right? That's most of your body. The special sex cells that we made, now do we make this during mitosis or meiosis? Sex cells, gametes? You're right, go ahead and say it louder, so. Yeah, meiosis. Alright, haploid cells have half the normal amount of DNA. Those are your reproductive cells. Yeah. Oh, darn, I had it on the board. Never mind. I'll stop asking you things I have on the board if I notice it. Anyway, we have one set of chromosomes, but that one set is not usually everything I got from mom that I have. So now let's look and make sure what you got from mom and what you got from dad. Because chromosomes line up with independent assortment. Remember that? All right. So we have a bunch of vocabulary words in genetics. We'll just fly through them really fast. A gene is instructions that are written in DNA. It's instructions to make a protein, and usually you're going to have lots and lots and lots of genes on a chromosome. The bigger the chromosome, the more genes you can fit on. But chromosomes tend to have lots of genes. Anyway, some of the words are really easy, really straightforward. The genotype is the type of genes you have. It's not hard. The phenotype is the things that you actually express, the type of things you express. So your hair color, your freckles, your blood type, your eyesight. Do you remember we had when we did our four-word problems, Paris? You remember the very first we were talking about squash plants, and we had yellow skin squash and we had white skin squash. Remember that? Well, the phenotype would be yellow skin, white skin. It's a description of the physical traits. It's not a description of what the genes are. Does that make sense? The genotype is what the gene is, where the phenotype is. Well, that's white skin. That's yellow skin. Oh, the watermelon is striped. That's a phenotype. Good? All right, so here we have a person. And she's got chromosome from mom, that's her maternal one. She's got chromosome from mom, that's her paternal one. Each of these chromosomes have the same genes. She's got two copies of A, one for each parent. Would you agree? Because it's the same chromosome, she should have the same genes. So she should have two copies of D. Does she have two copies of D? She should have two copies of A. But are they all the same version of that gene? No. Both of A are the same. So we're going to say that she is homozygous. Remember, homo means the same. We're going to say she is homozygous for A. Look at D. She's got a dominant on her dad. She's got a recessive from mom. She has not going to say that she's heterozygous. And then right here, we have two, they're both capital. So they're both dominant, but they're not the same thing. Do you see that? That's how your blood typing works. This would be a codominance where you have more than one dominant. Think of in your blood typing, A is dominant to O, but B is also dominant to O. And it's possible to have both the A and the B version of the gene. Then you would have what we call AB blood, right? That's what's going on right there. So, alleles are versions of a gene, and we're going to oversimplify in lines. There's only one gene involved in hair color. But in reality, there are several. But suppose there was only one gene for hair color. You'd agree that you could have black hair, or brown hair, or red hair, or blonde hair. Am I right? All right? Okay. You have different versions of that gene, different alleles. Same thing with our P gland. You've got the purple allele and you've got the white allele. They are the same gene, just different versions of it because we can do different things. When we talk in biology about genetic diversity, we're saying that you have more than one allele in the population. And we usually think that having more than one allele is a very good thing. The more alleles you have, the more chance you have that when the coming plague gets here, you'll have something that's immune to it. Does that make sense? If everything's inbred and too similar, then there's a chance that you'll die. Or if you don't die, there's a chance that you'll be weaker or come out looking weird. Sorry, I'll use my British royalty tips later. But you get my point, right? Right? Okay. So phenotype is the type of genes you are expressing, and genotype is the genes you have. We already did that. But we're going to break genotype down a little bit further. Homozygous means you have all the same alleles, all the same versions of the gene. Heterozygous means you have more than one version. Is everybody on board with that? Yep. Just nod, Javier. I know you know it. Okay, move on. Okay, so when we say hybrid in genetics, it doesn't necessarily mean this species within this. It could be within the same species. You had this lineage, this purebred purple flower, this purebred white flower, and we breed them together. What we get is a hybrid. Or what's the fancy H word for it? It's a heterozygote. Good, good. That's just my story. All right. If you got two different genes involved, and you're heterozygous for both, you're a dihybrid. Does that make sense? Yeah. All right. Straightforward enough. I want you to write these two things down carefully. Yeah, I know you've seen them. I want you to write them down. They are not the textbook definition of dominant or recessive. They are better because they work everywhere without having to modify. A dominant allele is the version that will be expressed if it is present.